The second proposed mechanism for ALS is the oxidative stress hypothesis. Oxidative stress causes strain on cellular function as a result of the body's inability to remove or repair the damage caused by reactive oxygen species. Reactive oxygen species are highly reactive molecules that are produced when tiny particles called electrons escape from the mitochondria during the energy-making process inside our cells. When these electrons escape, they can oxidize oxygen, causing it to change into these harmful reactive oxygen species. To oxidize, or oxidation, refers to a general process in which a substance loses electrons which can produce harmful molecules called free radicals. All molecules have electrons, but in order for the molecules to be happy, they need to have paired electrons. Free radicals are very unhappy and unstable because they have unpaired electrons, making them highly reactive chemicals that can harm cells. The two common types of reactive oxygen species are superoxide and hydrogen peroxide. Superoxide will combine with the free radical nitric oxide to form a damaging substance called peroxynitrite. Meanwhile, hydrogen peroxide will decompose into a harmful and reactive molecule called a hydroxyl radical. Reactive oxygen species are highly reactive compounds that can damage various cellular components, including cell membranes, proteins, lipoproteins, enzymes, hormones, and genetic material. Reactive oxygen species have also been shown to damage different transport mechanisms, such as the glucose transporters, which are specialized membrane proteins that facilitate the transport of glucose into the cell. The inability to maintain the balance of reactive oxygen species is often associated with a decreased ability of antioxidant enzymes to remove these reactive oxygen species. Antioxidants are the body's defense mechanism against free radicals. Antioxidants are molecules that are able to safely donate an electron to the free radical, allowing it to be stable and happy once again, preventing it from further cellular damage. An important antioxidant is superoxide dismutase 1. After analysis of post-mortem neuronal tissue, urine, cerebrospinal fluid, and plasma samples from patients with sporadic ALS, they found that there was a consistent oxidative damage to the proteins, lipids, and DNA, which suggests that oxidative stress is likely to be one of the cellular mechanisms of motor neuron degeneration. Moreover, specific markers for oxidative damage, such as levels of 3-nitrotyrosine, are consistently found in high concentrations in both motor neurons and cerebrospinal fluid in ALS patients with SOD1 mutations, as well as of sporadic ALS patients. Researchers have found more than 180 mutations in the part of the SOD1 gene that is responsible for making SOD1 protein in ALS patients. In addition, after scientists completely removed SOD1 in mice, there was no development of ALS-like symptoms. Therefore, rather than a loss of function, the mutated form of SOD1 is believed to have a harmful effect through a toxic gain of function. Moreover, mutated SOD1 proteins will misfold because they are damaged by oxidation. These misfolded proteins can be found inside the mitochondria of the spinal cords in mutated SOD1 mice. This results in an impaired energy production process in the mitochondria, causing increased susceptibility to oxidative stress and mitochondrial damage. Further, studies show that a higher oxidative state in cells promotes programmed cell death, which contributes to motor neuron degeneration in ALS. Except for radicava, which is believed to have antioxidant properties, other antioxidant therapies have shown less convincing benefits in affecting ALS. Vitamin E, which is the most active natural antioxidant in the body, delayed onset and slowed progression of the disease when fed to mutant SOD1 mice, but did not extend their survival. However, administration of N-acetyl-L-cysteine, an antioxidant molecule that alleviates free radical damage, through a slow injection under the skin to patients was not shown to slow disease progression or increase survival. On the other hand, studies on curcumin, a natural dye obtained from turmeric, increased survival of ALS patients and resulted in decreased ALS progression and a reduction of oxidative stress. However, the limitation of curcumin is its low oral bioavailability. In other words, only a small amount of it actually gets absorbed into the bloodstream, and also poor water solubility, thereby presenting significant obstacles to its delivery and effectiveness.